Growing up in Australia, we all would have seen this ad.
One damaged cell can start a melanoma growing. You haven't even started to burn yet.
Whilst this is good for raising awareness to slip, slop, slap, seek and slide, the aim of this video is to delve deeper into what skin cancer is, particularly melanoma. We'll also touch on the underlying pathophysiological processes that cause a melanoma, as well as the signs and symptoms that help detect a melanoma earlier, all whilst including some up-to-date statistics that help identify a need for awareness. That being said, let's dive right into it.
Generally, cancer is defined as the abnormal growth and proliferation of cells in an uncontrolled way. These abnormally growing cells have the potential to invade and damage surrounding tissues or spread to other parts of the body and cause damage. Therefore, skin cancer can be defined as the abnormal proliferation of skin cells. Our skin is divided into three layers, the epidermis, dermis, and hypodermis. We will focus on the epidermis as it contains the basal cells, squamous cells, and melanocytes, all of which correspond to the three types of skin cancers: basal cell carcinoma, squamous cell carcinoma, and melanoma. Basal and squamous cell carcinomas are more prevalent than melanoma, as evident by the 114,000 hospitalizations in Australia in 2013 and 14 compared to the 23,000 for melanoma. But melanoma has a much higher mortality rate at 6.2 deaths per 100,000 compared to 1.9 for non-melanoma skin cancers. For this reason, we will be focusing on melanoma. Normally, melanocytes function to produce melanin when exposed to UV radiation, like that from the sun. Melanin darkens the skin and helps dissipate UV radiation, therefore protecting us from damage. UV radiation causes the dimerization of pyrimidines. This may result in the activation of oncogenes, inactivation of tumor suppressor genes, and impairing of DNA repair. Let's look at how this causes melanoma in a bit more detail. Oncogenes form when a group of genes known as proto-oncogene become mutated. Normally, proto-oncogenes function to stimulate cell division, inhibit cell differentiation, and halt cell death. Therefore, when mutated to an oncogene, it causes increased cell division, decreased cell differentiation, and inhibition of cell death. In melanoma, the genes mutated are BRAF and NRAS, which relay signals to proliferate and differentiate, therefore causing a cancer to grow. But tumor suppressor genes act to prevent this rapid and uncontrolled division. The CDKN2A gene encodes for the P16 INK4A and the P14ARF proteins, which stop cell division in older cells by binding to and blocking the CDK4 and CDK6 proteins that normally signal to progress through the cell cycle. Furthermore, P16 INK4A also protects the breakdown of P53, another tumor suppressor gene, therefore preventing tumor formation. When the CDKN2A gene becomes mutated, there is no signal to stop tumor growth, therefore allowing the uncontrolled proliferation and differentiation of melanocytes, resulting in a melanoma. Let's look at this on a cellular level. To begin with, a benign melanocytic nevus, known as a mole, develops due to controlled proliferation of melanocytes. A BRAF mutation occurs, resulting in uncontrolled growth of melanocytes characterized by an atypically shaped nevus with irregular borders and different colors. Melanocytes then acquire a CDKN2A tumor suppressor gene mutation, which allows melanocytes to spread in the epidermis. E-cadherin confines these cells to the epidermis, but when CD1 is increased and other biochemical reactions occur, E-cadherin is lost and melanocytes can invade the basement membrane and infiltrate the dermis. Finally, a metastatic melanoma can develop when they enter the bloodstream and spread to other tissues and organs. This process can be avoided with early detection of the signs and symptoms.
These are as easy as learning your ABCs. A melanoma is asymmetrical and has a non-uniform border. Melanoma often has different colours, like brown or black or even red and blue. Often the melanoma will have a larger diameter than a pencil eraser and will evolve over time. This links back to pathophysiology. Remember, melanocytes produce melanin which stain the skin dark. If there is abnormal growth, then there will be abnormal staining, like what I just mentioned.
If that hasn't convinced you to put on some sunscreen, then maybe some of these will. To start with, Australia and New Zealand have the highest rates of melanoma in the world, at 11 times higher than the world average. It is Australia's fourth most common cancer, with 1 in 13 and 1 in 22 women being diagnosed. The majority of these are those aged 60 to 69, but the highest incident rate belongs to those over 80. Indigenous Australians have a lower risk due to their higher melanin concentration, causing their skin to be darker. This is represented by them only contributing to 0.2% of melanomas at a rate of 9.3 per 100,000 compared to 35 for non-Indigenous Australians.
So when you're out enjoying our beautiful sun, don't forget to listen to Mum and protect yourself from the harsh radiation by slip, slop, slapping. Those ads are there to help you, and I hope that this video has helped you better understand why.